Hello again, Year 8. Let's just open that slideshow for you. There we go. Okay, so title, just move me out of the way. Title for this part of the session is Osmosis. So we're going to be looking at what osmosis means and comparing it to diffusion. So please get your keywords written down: water particles, partially permeable, concentration, gradient, and cell membrane. And then I'd like you to think about what we mean by the term specialized cell. And what is the function of chloroplasts? So your teacher might want to pause the video for a few minutes just to let you get all of that done. Okay, so the keywords are very similar to the ones we used in the diffusion lesson, so I'm not going to go through them all one by one, but we'll recap them as we go through the lesson. So our do now task, specialized cell. Well, hopefully you remembered from our previous lesson that specialized cells are cells that are adapted for a specific function. So, for example, the function of a sperm cell is to fertilize an egg, and to do that it has to get to the egg, so it has a tail for swimming. So the function would be to fertilize the egg, and the adaptation would be tail for swimming. Another example would be a red blood cell. It is donut shaped to give it a bigger surface area, and that is how it is adapted for its function, which is to carry oxygen around the body. Next question, what's the function of the chloroplasts? Well, the chloroplasts are those little green blobs that we see pictured inside a plant cell, and their job is to absorb light energy for photosynthesis. And to remind you, photosynthesis is a chemical reaction that plants carry out. It's how they make their own food, and it also produces oxygen, which of course is really useful for all living things. Okay, so moving on. So, osmosis. I'm going to show you a video now. I'm going to take my face off the screen while the video's on, and I'm going to show it to you. It's a video about osmosis, what it is, how it compares to diffusion, and also some examples of osmosis in living things. Let's get rid of me. There we go.
You know what word gets thrown around a lot, but is actually pretty challenging to understand? Osmosis. But it does answer some great questions like, why would it be deadly to give someone an IV of pure water? Or why would a saltwater fish placed in fresh water experience major problems? We'll answer these questions as well as explain the process of osmosis. Now first understand that osmosis is the movement of water, and that really is a type of diffusion. Often when you're talking about osmosis, you're talking about the movement of water through a semi-permeable membrane like a cell membrane. A cell membrane has openings small enough for water molecules to pass, but will not allow larger things like salt molecules to freely pass, at least not without assistance. Osmosis is a type of passive transport, meaning that it doesn't require energy. I mentioned osmosis is a type of diffusion, and similar to what you might have learned about with diffusion, molecules, in this case water, travel from areas of high concentration of water to low concentration of water. But there's another way to think about water movement in osmosis. A low water concentration likely means there are a lot of solutes. Solutes are substances like salt or sugar that can be dissolved within a solvent, like water. Water has the tendency to move to areas where there's a high solute concentration, which means there's less water concentration. So if you want to easily figure out where the water will travel, look to the side where there's a greater solute concentration. So let's imagine you have a box with a semi-permeable membrane in the center. And remember, this membrane will let tiny things pass through it, like water and gases, but not large solutes like salt. We'll label one side of the box A and one side of the box B. If you have pure water on both sides of the box, those water molecules, they're moving. Water molecules, they like to move. But the net movement of water on the two sides is zero, meaning the overall change in direction of movement is zero. Now let's imagine on side B, you dump a huge amount of salt there. So which direction will the water move towards? A or B? The answer is B. This area has a high solute concentration. And remember, water moves to areas of higher solute concentration. You can almost think of the water as trying to equalize the concentrations, diluting side B. Once equilibrium is reached, the net movement of water on the two sides will be zero. But remember that water still likes to move, and movement still occurs. Here's some vocabulary to know. We call side B hypertonic. You know that prefix hyper when you think of a hyper person? Well, that helps me think of a high amount of something. For a hyper person, it might be caffeine. But in our case, it means a high solute concentration. But we can't just say something is hypertonic without comparing it to something else. We say that side B is hypertonic to side A because it has a greater solute concentration than side A. In osmosis, water moves to the hypertonic side. We say side A is hypotonic. Hypo rhymes with low, which helps me remember that it's a lower solute concentration, at least when compared to side B. So let's do some real-life examples now, because this box thing is getting a little bit boring. When someone gets an IV in a hospital, it may look like the clear fluid running through the IV is water. But it is certainly not water. That would actually be a disaster because of osmosis. Let's say hypothetically there was pure water placed in an IV. Now, an IV tube typically runs through a vein, so that you have access to your bloodstream, really useful for running medication through. Blood actually consists of many different types of cells. Red blood cells are a great example. So what do you think has a greater solute concentration? The hypothetical pure water in the IV tube or in the red blood cells? Remember, cells are not empty vessels. They do contain solutes. The pure water that hypothetically is running through this IV tube has no solutes. It's just pure water. So where does the water go? Remember, it goes to the area of higher solute concentration. So it's going to go inside the cells. The cells are hypertonic compared to the water in the IV tube because the cells have a greater solute concentration. The cells would swell with water and possibly burst. Exploding red blood cells are not fun. If a person needs fluids, they typically receive a solution that is isotonic to their blood plasma. Isotonic means equal concentration. So you wouldn't have any swelling or shrinking red blood cells. And that's a good thing. Say you go to a pet store and you get a fish that looks like Nemo from the movie Finding Nemo, a clownfish, only you don't know it's a saltwater fish. What would happen if you placed this saltwater fish in fresh water? Okay, first ask yourself, where is there a higher solute concentration? In the saltwater fish cells or in the fresh water that you just placed this fish in? Definitely in the saltwater fish cells. So where does the water go? It's going to go to the area where there's a greater solute concentration, the hypertonic side. So it's going to go into that poor fish. If you don't rescue it soon, the cells would actually start swelling if you looked at them under a microscope. Eventually, this fish could die. Now, one thing to clarify, saltwater fish and freshwater fish are not necessarily isotonic. Remember, that means equal concentration to their surroundings. But they have special adaptations that allow them to live in their environment, and usually they can't make a major switch from a saltwater environment to a freshwater environment. Now, not all fish have this problem. There are some fish that have this amazing adaptation to switch between fresh and salt water, and they have to deal with this osmosis problem frequently. Salmon, for example. I think if I had to be a fish, I'd be a salmon. Osmosis also explains how many kinds of plants get their water. Sure, many plants have roots, but how does the water get into the roots? When it rains, the soil becomes saturated with water. The root hair cells have a higher concentration of solutes within them than the solutes in the saturated soil. So the water travels to the area that is hypertonic, and in this case, the roots. Or if you live in an area that gets ice, you might be used to salt trucks salting the road. Great for icy roads. Not so great for the plants that live right by the roadside. If you dump a bunch of salt on these plants, you can make the soil environment around them very salty, very high in solute concentration, very hypertonic compared to the root hair cells. So how does the water move into the root hair cells now? Well, it doesn't. Many plants by salted roadsides die. The same kind of thing can be tragic for slugs. We think slugs are misunderstood, almost cute gastropods. We understand that many gardeners find them to be pests in their garden, but we really don't like it when we hear of salt being placed on slugs. It really is death by osmosis. Basically, if salt is dumped on a slug, you are making that outside environment of the slug right there next to it very hypertonic. The slug is hypotonic to the salt. Remember, the water will travel to the area of higher solute concentration, to the hypertonic side. So the water that was in the slug, the moisture that this slimy slug needs, travels out of the slug, and the slug dehydrates. You can get really technical with osmosis. In advanced biology, you explore a formula to calculate water potential, and that in osmosis, water travels to areas of lower water potential, basically areas of lower free energy. We won't be going into the water potential calculations with this short clip, but we like to think about water as loving solutes and avoiding pressure whenever determining where will the water go. Water is one of the most important molecules for life. So the big picture here is in all topics of life, it is really important to understand how water travels. Practice makes perfect, and so we have some practice problems if you'd like to test out your osmosis skills. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, I hope you enjoyed that. I am just going to try and put myself back on the screen. No, that's not going to work. Never mind. You can manage without my face on your screen, I am sure. Okay, so these were the things. Sorry, folks, I'll just get this sorted. So these were the things I asked you to look out for. What osmosis is, why it's a passive process, and the direction of the concentration gradient. So hopefully you cottoned on that osmosis is very like diffusion. In fact, it's a special type of diffusion. Osmosis is diffusion of water. It's movement of water molecules from a high concentration of water to a low concentration of water. And the video introduced some new keywords which we didn't write down at the start of the lesson because I forgot to put them on the slide. Hypertonic and hypotonic, so I'll come to those in a moment. It's a passive process because it doesn't require energy. And as with diffusion, we're looking at a concentration gradient from high to low. But this time we're looking at high concentration of water to low concentration of water. because osmosis is basically diffusion of water. I'm going to skip past that. This is a definition that you can now write down in your books, please. Osmosis is the movement of water molecules from an area of higher concentration of water to an area of lower concentration of water through a semi-permeable membrane. And if you were writing that in a GCSE paper, think about which are the most important bits. What would you get marks for if you wrote that brilliant definition in a GCSE paper? So the video could be paused now for you to finish that off. Okay, I'm going to move on now. So if you're not finished, the video does need to be paused now. So these are the most important bits. What's really key about osmosis is that we're talking about movement of water molecules specifically. If it isn't water moving from high to low concentration, then it's not osmosis. We would just call it diffusion. High to low concentration is also very important. You could also say down a concentration gradient, and the semi-permeable membrane is another good point to always make as well. Okay, so these key words that were mentioned in the video. An isotonic solution, if we have an isotonic solution outside of a cell, then we mean that the concentration of water and solutes is the same inside and outside the cell. So the word solutes just refers to anything that is, do you know what, I'm going to try and get my camera working again quickly. Sorry about that, folks. I messed up my recording. Let me just get you back. Okay. Right, I'm just going to also move me. You don't want me in the middle, do you? Hello. There we are. Right, so we were talking about different types of solutions mentioned in our video. Oh, that's not much good either, is it? Perhaps it was better off down here. Here we are. Okay. So isotonic solution, if we've got an isotonic solution outside of a cell, that means it is the same concentration as the solution inside the cell. So solutes are salts and sugars. So if we've got an isotonic solution outside of the cell, that means the solution has got the same percentage of salts or sugars and water as the liquid inside the cell. So you wouldn't be getting any osmosis because the concentrations inside and outside the cell are exactly the same. Hypertonic solution has a higher concentration of solutes, so that's the salts and sugars, than inside the cell. Okay, so imagine you've got fluid outside of a cell. The cell has got some sugars and salts dissolved in it, but the fluid outside of the cell has got more sugars and salts dissolved in it, so it's hypertonic. So if we've got a high concentration of sugar and salt, that also means we've got a low concentration of water. If you think about your drink of squash, if you make a really, really strong glass of squash, then that's got a low concentration of water. If you then add lots of water to it and make it really, really weak, it's now got a higher concentration of water. So hypertonic, lots of squash or lots of sugar and salt would mean a low concentration of water. Hypotonic means a high concentration of water. So that's your weak squash or your solution which has got just a tiny amount of sugar or salt in it. Hypo means small amount of sugar or salt, but actually that means we've got a large amount of water, so a high concentration of water. So this can be a bit confusing. It's easy to get your hypo and your hyper mixed up. So I'd like the video to be paused now just for a few minutes so that you can get that written down so that you've got it to refer to. The really important thing to remember is that hypertonic, in fact, I'm going to take out to make it a little bit less writing for you. We'll just say hypertonic solution. The solution has a high concentration of solutes, so low concentration of water. And for hypo, the solution has a low concentration of solutes. I'm just going to correct that too. Sorry, I remember now that I meant to do this before I record the video. There we go. That's a bit less for you to write down. So isotonic, hypertonic and hypertonic. And if you get that done quickly, I want you to think about which way would water move if you had a hypertonic solution outside of the cell. Which way would water move? Would it move into or out of the cell? And if you had hypotonic, would it move into or out of the cell? So video pause now and get that written down. Okay, so hopefully if you had a go at challenge, you realised that with a hypertonic solution outside of the cell, so a low concentration of water, water will move from inside the cell outwards towards the low concentration. So that's usually a bad thing because usually we want our cells to contain water. With a hypotonic solution, so a weak solution if you like, we've got a high concentration of water outside of the cell and water will move into the cell. Now usually that's a good thing because usually we want our cells to be hydrated. But if you have pure water outside of cells, then so much water moves into cells that cells can burst, and that was shown on the video that you just watched. The example of the IV drip, when we give people a drip into their blood vessels to give them water or blood, then the solution we give them has to be isotonic or hypertonic, not hypotonic. And that's the movement of water there, hypo to hypertonic. So a quick summary then of osmosis, some key points. Osmosis in plant and animal cells is different because of their different structures. I just talked about animal cells bursting potentially. That's less of an issue with plant cells because they have a cell wall. Do you remember that plant cells have a cell wall as well as a cell membrane? Animal cells don't have a cell wall, they just have the cell membrane, so they're much more fragile. Plant cells are a bit tougher, and if they absorb a lot of water, it's not such a problem for them. So animal cells will burst if they take on too much water. They'll shrink if they're lacking water. When plant cells lose water, they don't shrink as such. They become what's called plasmolyzed. So the inside, it's sort of the inside of the cell that goes all floppy. The vacuole pulls away from the inside of the cell. So the cell kind of gets floppier rather than smaller. So here's an example for you to think about. And this is the last thing you're going to do in this lesson. So I want you to think about why somebody might go on holiday and when they come back, their plant that looks really, really healthy now looks really, really wilted and unwell. And the next thing I want you to think about is your tears. Tears have a high salt content, and that basically causes bugs and bacteria that might get into your eyes to dry up. So think about why that might happen. It's a bit like the slug example that you saw in the video. Okay, so I want you to, depending on how much time is left, you could have a go at both of these or pick one. And it would be easy, wouldn't it, to say, well, the plant's dried up. But I want you to think about what's happening in terms of osmosis. The plant hasn't been watered, so there's a low concentration of water in the soil. So what might happen? And also what might be happening to these microorganisms in your salty tears. So if there's time, the video can be paused now and you can have a go at that. Okay, so let's have a look at these examples. So when the plant is healthy, it's being watered regularly, there's a relatively high water concentration in the soil. And so if the cells are getting dry and have a low concentration of water, then water will move across the cell membranes from a high concentration of water in the soil to the low concentration of water in the cell by a process called osmosis, which is diffusion of water. But when that soil starts to dry up because it's not being watered regularly, we've got a lower concentration of water in the soil than we have in the plant cells. We've now got higher concentration of water in the cells and a lower concentration of water in the soil. Which way is the water going to move by osmosis? Out of the cell into the soil from an area of high to low water concentration, and that causes those cells to dry up and become plasmolysed. That was the word that you saw. You don't need to remember that one, it's just quite a cool word. Plasmolysed, when the vacuoles of the cells kind of shrink down. And your tears, well, if tears have a high salt content, then it's a bit like putting salt on a slug. We've got a lower concentration of water and a high concentration of salt in our tears. Our microorganisms have a high concentration of water and a low concentration of salt. So water will move by osmosis out of the cells, out of the bacteria and the bugs into, outside of the cell into your tears. So they sort of dry up and dehydrate, just like when you put salt on a slug. Please don't try that, it's not very nice. I know slugs aren't very nice, but we should be kind to slugs. It's not their fault they're ugly and slimy, is it? Just a thought. Okay, that is the end of the lesson. If there's anything you don't understand or you've got any questions, then it might be an idea to make a note of them at the back of the book so that when you're back in school, if you're doing this at home, or when I'm back in school, if you're watching me from the classroom, we can go through your questions together. Okay, bye-bye.